Welcome to DDHS News, the place where we look at real biomedical research while the rest of the internet argues about whether celery juice can fix your mitochondria. Today we are going into the good stuff, the data, the discoveries, the generally mind-bending clues scientists are uncovering about neurodegenerative diseases. Because this week, the brain stole the spotlight. Researchers around the world have dropped some major insights into how Alzheimer and other neurodegenerative diseases develop, from early molecular signatures to weird protein deletions to how your heart health might literally shape your brain. So grab your coffee, settle in, and let's explore what the science actually says. No hype, no pseudoscience, just a fascinating truth. Hi guys, welcome back to Daily Dose of Honest Science. I'm really happy to have you back. Today I'm trying a fresh new format called DDHS News. So you're not weekly dose of science, you didn't know you need it, but your neurons will definitely appreciate. Before we start, just a very quick note. Dear Mr. Stephen Colbert, dear CBS, please don't strike my video. I'm borrowing the format, I'm not stealing it, and since you're leaving soon, consider this as a tribute. From a science nerd, let's say, to a late-night royalty. Because yes, just like Stephen Colbert's, meanwhile, we will be taking some detours into scientific weirdness happening behind the scenes. And honestly, science is wild, confusing, and occasionally hilarious, so I think we should embrace that. And today we're diving into 3 brand new papers on neurodegenerative diseases, so Alzheimer, frontotemporal dementia, and CTE, and of course all the fun stuff you probably wouldn't learn from late night TV. So let's get started. Let's start with a very recent paper with the complicated title I Will Spare You. It got published in October 2025 in the Journal of Cardiovascular Research and investigating an all-time favorite hypothesis: do people with cardiovascular diseases bear an increased risk for dementia? Well, epidemiological studies, I guess I first explain what that is. So epidemiology is like basically a detective work for medicine. It tracks who gets sick, how often, and why. Think of epidemiologists like Sherlock Holmes for public health, just with fewer pipes and smokes and a lot of more spreadsheets. So as I said before, epidemiological studies showed that patients affected by cardiovascular diseases do bear maybe an increased risk for dementia, but the potential mechanism underlying the interplay are still not really understood and found also not demonstrated in clinical data. So the authors thought, why not getting insights? They specifically focused on so-called allostatic interoceptive network, or short AIN. Well, how can I explain now AIN? AIN is like your body's high-tech control room. One system checks what's happening inside and the other fixes it before even something gets noticed. So it's like the brain's version of 24/7 maintenance crew, making sure that the inner thermostat, the fuel gauge, or stress alarms don't go all off at once. So why would you investigate that? So what's kind of the question or hypothesis you want to have? First of all, you want to understand the growing interest in how cardiovascular health impacts brain health and neurodegeneration. So the authors hypothesized that common cardiovascular risk factors like hypertension, lipid levels, and metabolic stress contribute to an allostatic interoceptive burden, so like this internal stress load. And that could accelerate neurodegenerative processes. And the study aimed to quantify these associations between cardiovascular risk factors and this allostatic markers in humans to test if cardiovascular risk should be integrated into models of neurodegenerative diseases. What kind of methods they actually used? So the researchers used an observational cohort data. So they collect data on cardiovascular risk factors and measured markers related to allostatic or interoceptive load. And the matrices likely involves physiological biomarkers. And the advantage of this approach is it allows investigating in real human populations, not just in animal models, and capturing the variability in risk factors exposed in internal stress load, giving translational relevance. What was the main finding of this paper? Well, the study found that there is a significant association between cardiovascular risk factors and elevated allostatic and interoceptive burden. So that kind of means that people with worse cardiovascular risk profiles tend to have higher internal physiological physiological stress load. And this kind of supports the idea that cardiovascular health and the internal stress are linked in ways that might affect a long-term neurological health. So the authors argue that these results kind of underscore the importance of integrating cardiovascular health into models of neurodegenerative diseases. We also need to talk about the limitations. And as the authors note, this is an observational correlation study. So it means association does not prove causation. We cannot conclude that cardiovascular risk causes increased allostatic burden or neurodegeneration. The allostatic or interoceptive burden as an index, but its components, reproducibility and long-term predictive value for neurodegenerative outcomes remain really uncertain. So the study sample size, the participants' demographics, and the potential confounders like lifestyle, genetics, or other illnesses are not clearly defined in the summary, which may limit generalizability. So we need longitudinal follow-ups, also neurodegenerative disease onsets, and apparently it is not part of the current work, so it's unclear whether elevated allostatic burden indeed translates into higher neurodegenerative risks. So what's the overall conclusion and why does the study even matter? Well, in short, this work highlights a potential underappreciated link between cardiovascular risk and internal physiological stress load. So bridging kind of the cardiovascular and neurological research field. It suggests that beyond traditional endpoints like heart attack or stroke, cardiovascular risk may also contribute to brain health via chronic internal stress load. For you guys, this study is important because it broadens how we think about cardiovascular risk. So it's not just about the heart disease risk, but also is now a factor relevant for neurodegeneration and brain aging, therefore underlining the need for holistic health maintenance. So yes, taking care of your heart may literally help protect your brain. Cardio isn't just for fitness, it's for cognition. The study reinforces taking care of your cardiovascular system is basically taking care of your brain. Meanwhile, somewhere out there, a treadmill is being used exclusively as a clothes hanger. Science calls this sedentary lifestyle. Interior designers call it multifunctional furniture. What if we could read the molecular fingerprints of aging in people who are still cognitively normal and use that information to catch Alzheimer's before it's even visible at start? That's exactly what this new study attempts by profiling thousands of proteins in the cerebrospinal fluid and uncovering patterns that hint at neurodegenerative risk. So this paper is more likely an early proteomic signature in CSF long before Alzheimer's symptoms actually appear. So if I would give a one-sentence summary, it would be this study uses deep proteomic profiling of cerebrospinal fluid, so-called CSF, in cognitively normal people, the ApoE4 genotype, and early signs of Alzheimer's-linked neurodegeneration. So how was the study actually conducted? We had 994 cognitively normal adults as participants. The age was between 43 and 91 years old. Groups or factors that were analyzed instead of predefined disease groups, the study looked at how protein patterns in the CSF vary with age, with sex, and the ApoE4 genotype, which is the major genetic risk factor for a developing Alzheimer's disease, and the overall amyloid status, so presence of amyloid as an early Alzheimer marker. These factors were analyzed individually and in combination to see how they shape the proteomic landscape. Now when we look at the main results, what did they figure out? Well, first of all, the proteomic signature. So the authors identified thousands of proteins whose level were associated with aging, sex differences, the ApoE4 carrier status, and the amyloid positivity. Then, of course, there was an extensive overlap suggesting that these biological factors somehow interact in shaping brain-linked proteomic changes even before cognitive impairment. Then they did some network analysis. You have to imagine each protein can be associated with a certain network or a certain pathway in the brain, or in general to any kind of signaling pathway. By grouping proteins into a co-expressed module, they found like clusters linked to neuropsychiatry, inflammatory and aging-related pathways, and therefore offering potential early indicators of neurodegenerative processes. So overall, the study has shown that the CSF proteome has unique aging signatures distinct from the plasma proteomics. Okay, what are the limitations of this study? Well, it's an observational and cross-sectional study. Because the data comes from a single time point per person, it cannot really establish like a cause effect or predict the true progression of Alzheimer's. The cohort representativeness is also a bit the limitation, because a small sample size, although being nearly 1,000 participants, the sample may not really fully reflect the genetic, ethnic, or lifestyle diversity worldwide, which could also affect their generalizability. Then, of course, the biological interpretation. While the study reveals association, the functional role of many of these protein changes are not really clear and also not known how they would drive the neurodegeneration. Another limitation from my side is the scan that they use. They use the so-called SomaScan 7K platform. So on a quick footnote, this study didn't use mass spectrometry, so the instrument that I'm working with, they used an aptamer-based platform, which means it's great in scale, but comes with different trade-offs. First of all, what are aptamers? So these are like little synthetic DNA or RNA binding hooks, you could say. They latch onto a specific protein that has been designed for. You could imagine this like a nucleic acid version of the antibodies. What can the SomaScan do well? Well, it measures a predefined, in this case, 7,000 protein at once with really large cohorts. And of course, it's very sensitive and for especially low abundant proteins in fluids like plasma or CSF. And since it's a platform that has been designed with these 7,000 predefined proteins, it's highly reproducible and scalable. And that's why it's making it so popular for biomarker screens and epidemiological studies. But we also need to keep in mind what kind of limitation that has. Since the SomaScan only has this fixed panel, other proteins or isoforms or any modification on these proteins cannot be included. So they kind of remain invisible in the assay. And sometimes the signal also depends on how these aptamers bind to the protein. And sometimes it reflects how accessible the epitope, so like how the recognition site of these aptamer to the protein is, and does not really truly sometimes get into the true protein abundance. It provides one signal per target with no direct information on the isoform, or if this is maybe a cleavage product, or maybe if it has been modified, so it has been decorated with some chemical groups. So what can LCMS, the main thing that I'm working, offer instead? Well, LCMS can be more unbiased. It detects actually proteins without pre-selecting the target. It gives a sequence level information, allow us to see isoforms and many, of course, PTMs. Therefore, makes LCMS better suited for deep mechanistic insights, not just like pattern detection. So why does this matter now in CSF? Well, CSF contains a lot of low abundance proteins. In this case, only 7,000 has been measured. It could be that in CSF we have specific proteins or proteoforms or different kind of proteins that have been decorated that you miss with the SomaScan. Great way would be to measure it with the SomaScan, but also have in addition some insights with LCMS, so you complement each other's assays, so you get the most, let's say, detailed insight. So this fixed aptamer panel might miss important biology or collapse multiple forms of one protein in one single signal. So the results are powerful for relative comparison, but they don't really represent a complete mechanistic proteome. Overall, the SomaScan is excellent for large-scale screening, but the LCMS goes deeper. So especially when we want to understand like mechanisms, isoforms, and modifications. And also the signaling itself, so signaling pathways can also be uncovered by this. What's the overall conclusion and why does this study actually matter? Well, this paper expands our understanding of the earlier molecular changes in the brain years before Alzheimer's disease becomes clinically apparent. By identifying CSF protein signatures, linking it with age, genetic risk, and early pathological markers in people without any symptoms, it helps to bridge the gap between normal aging and early disease biology, potentially guiding future biomarkers and interventions. The study nicely builds on work I have covered previously in a podcast episode. It's a work that has been published in January 24 in Nature Aging that used the CSF also and made also proteomics with that to define 5 molecular subtypes of Alzheimer's disease with distinct genetic risk profiles. And I highly recommend watching that episode for context. So these early protein shifts may help us to identify Alzheimer's long before symptoms actually appear, and maybe even before we realize we have lost our keys for the 5th time this week. Meanwhile, somewhere on the internet, someone is still convinced Alzheimer's is caused by aluminum foil. Don't worry, this is not true and complete nonsense. So CSF proteomics gives us an early warning sign, like a molecular whisper before Alzheimer becomes visible, but the next paper asked a much more deeper question. What if the disease process is already written into the DNA of neurons themselves? That brings us to the final study. If I now again give a short summary in just one sentence, the study is showing us that neurons in chronic traumatic encephalopathy, short CTE, accumulate unusually high levels of DNA damage and somatic mutations far beyond normal aging, revealing CTE as a disease with a strong genetic component, similar in key ways to Alzheimer's disease. So what's the background? Why is it important to investigate? So CTE develops years after repetitive head impacts, RHI, especially in contact sports, but it has been shown that not everyone exposed to RHI develops CTE, suggesting there's like an additional disease mechanism that we have not fully understood. So CTE and Alzheimer's disease both involve tau pathology, yet their underlying molecular drivers are still poorly understood. And somatic mutations, DNA changes that accumulate over lifetime, even in dividing neurons, increase with age and are elevated in Alzheimer's disease. So the key question, do neurons in CTE show disease-specific DNA damage pattern that could help explain why some individuals develop CTE while others not? What kind of methods did they use? So researchers used single-cell whole genome sequencing. So they analyzed like the whole genome of a single person in a single neuron rather than taking like the bulk brain tissue. And these are two specific sequencing approaches. They are each other complementary. So one is called a strand-agnostic method to measure total somatic mutations, and the other one is strand-specific duplex method to distinguish the single versus double-stranded DNA damage. So the question is, why does this matter? Well, single-cell genomics allows detection of rare neuron-specific mutations that would be completely missed in a standard sequencing. So that's why it's so critical for understanding neurodegeneration. How's the actual study design? Participants, group, key factors? Well, they use brain tissue coming from the dorsolateral prefrontal cortex, or just from that part here. Groups analyzed were 15 individuals with neuropathological confirmed CTE, 4 individuals with repetitive head impacts but without CTE, 7 individuals with Alzheimer's disease, and 19 neurotypical controls. Key factors that now were analyzed are, as I said, somatic single nucleotide variants, explain the somatic and single nucleotide variants, then of course insertions and deletions, mutational signatures linked to aging, oxidative stress and DNA repair pathways, also associating it with age, symptom duration, APOE genotype, and of course years of football exposure. So what are the main findings of this study? Well, neurons from the CTE brains contained a roughly 114 extra point mutations per cell, comparable to Alzheimer's disease and well above normal aging. Importantly, RHI alone did not increase the mutational burden. Individuals exposed to head impacts without CTE actually looked like normal healthy controls. We also see axis mutations in CTE, which were driven by mutational signature C, a pattern that's also seen in Alzheimer's disease. And the signatures linked to like oxidative stress and impaired DNA repair, suggesting a shared disease pathway between CTE and AD. Some CTE individuals showed hundreds of axis small DNA deletions per neuron, equivalent to more than if someone would be 100 years of normal aging. These, let's say, high indel CTE cases closely resemble a similar subgroup of Alzheimer's patients. So they figured out that there's a specific DNA damage mechanism underlying there. So there's a dominant deletion pattern called ID4, and that one has been linked to topoisomerase 1 mediated DNA damage, a process that can occur during transcription. So our cells are capable of having different pathways to repair actually DNA damages, and one of them is through topoisomerase 1. So usually topoisomerase 1 we need as an enzyme to, you have to imagine the DNA is very twisted, right? And there's a lot of tension on the DNA. And if we don't release the tension during transcription, so if we want to make from a DNA an RNA, then it could be that also breaks happen there. So topoisomerase 1 helps to get kind of relax a bit the DNA, but also is involved if there's a strand break that it helps to recruit specific proteins that can repair this DNA then. And on top, this suggests that highly active neuronal genes may be especially vulnerable, leading to a cumulative damage in essential neuronal pathways. These are interesting findings, but we also need to talk about the limitations. Well, the study is post-mortem and an observational one, so it cannot prove any causation. And the RHI without CTE group was small and had a narrow age range. Most CTE cases were advanced diseases between stage 3 to 4, so early disease mechanisms remain unclear. No somatic mutations were found in classic neurodegeneration genes like APP, MAPT, or ApoE, suggesting that the DNA damage is distributed across the genome, not driven by single genes. What's the overall conclusion, and why does the study even matter? Well, the study reframes CTE as a more than mechanical injury disorder. It is a disease marked by progressive transcription-linked genomic damage in neurons, closely overlapping with mechanisms that we see in Alzheimer's disease. For you guys, as a takeaway, long-term neurodegeneration may depend on how neurons actually respond at the DNA level, opening new avenues for biomarkers, risk stratifications, and potentially shared therapies across tau-based neurodegenerative diseases.
Meanwhile,
somewhere in the genome, let's delete something important. What could go wrong? DNA is the chaotic neutral of biology. So that's DDH news for today. 3 papers, 3 insights, and 3 slightly confusing biological systems trying their best. If you enjoyed it, please hit subscribe, leave a comment, or send this video to someone who really loves science or just needs a reason to do some cardio. Well, we see us the next time. Same neurons, new news, and remember, as always, let a little dose of honest science into your life and brighten up your day.